Hello, my name is Andrew Brown. Today I'm going to be sharing with you a portion of my doctoral defense. The reason I would like to show my doctoral defense and post it here on the STEM Center's YouTube channel is because when I was originally planning on embarking on this PhD journey, I was looking for different resources that would show me a little bit about the type of research that's conducted in a scientific lab, in a PhD lab, how that research is conducted, like the logic behind making those experiments, how much autonomy do you have in your own research? So essentially trying to understand like how much ownership do you have in your own research. And also I was looking for resources that would show how to go about embarking in scientific discussions in like a public setting. So I figure if I post this here for those people that may be interested in doing a PhD, it might give them the opportunity to determine if this is something they'd like to do by seeing sort of the tail end results of a full PhD. So without further ado, this is a portion of my doctoral defense in biochemistry.
His thesis and things around it, and then we bring him back in, and that's the logistics. So, with that said, I'm going to turn it over to Andrew to begin, and then we can get going.
Okay, sounds great. Hello, my name is Andrew Brown. I am originally from Atlanta, Georgia, but I am the child of two immigrants from Liberia, Africa. I will be presenting to you today my doctoral research towards point-of-care diagnostics to monitor inflammatory biomarkers. And all of this work is done under the guidance of Dr. Suri Iyer. So essentially, the goal of our research is to, we develop assays that will eventually be translated into point-of-care tests. So you can imagine your average pregnancy test or COVID-19 test. So we develop assays in the lab that will eventually be translated. So the current protocol for individuals that become sick is they get a disease or some sort of chronic infection. They'll visit the hospital. They'll provide samples for the hospital to be analyzed. Those samples will be sent over to a lab, and that lab will analyze the samples and give that data over to the physician, in which the physician can go ahead and modify the dosage according to the patient's needs. So maybe they'll increase the dose of a certain drug or change a certain therapy depending on the disease state of the patient. So what we're proposing is a daily or hourly self-testing, self-monitoring at home so that the physician can get as much data as possible and as quickly as possible. So in our protocol, you get the disease or infection, just like before. You incubate with some sort of sample, either saliva, urine, or some sort of other blood sample. You incubate with some sort of signaling molecule. So in our case, we typically use nanoparticles as the quote-unquote transducer or signaling molecule, but it could be any signaling molecule. You record the data, and this process happens very quickly, usually minutes to hours, where you'll get the signal and be able to get data. You'll communicate with your physician, and your physician will be able to remotely modify the dosage. So again, we are developing assays that will eventually be incorporated, ideally, to point-of-care tests. So these are the main projects that I was working on in my lab in Dr. Suri Iyer's laboratory. So in the first project, I was working on assays to detect and quantify the cytokine interleukin-6. So we'll talk all about that. In the second project, I worked in collaboration with Dr. Mukesh Kumar's group to monitor dengue virus in patient samples. So we monitored both host biomarkers, so biomarkers that are produced by the body, and viral biomarkers, so pieces of the virus itself. However, I'm mainly going to be focusing on the assay development for the host biomarker, monocyte chemoattractant protein 1, or MCP1. And all of this work is published in ACS Bioconjugates if you enjoy this talk and you want to go read more about it. In Project 3, I transitioned to monitoring the capsid protein P24 using a layer-by-layer approach with site-specific and homogeneous antibody conjugates. And I'll explain a little bit more about what that means momentarily. Okay, so in Project 1, without further ado, towards point-of-care diagnostics to monitor the inflammatory biomarker interleukin-6. So essentially, interleukin-6 is a cytokine, so that means it's a signaling molecule, and it's involved in cell-to-cell signaling, so it allows cells to communicate with each other. You can see interleukin-6 right here. It's a 4-helix bundle. It's about 21 kilodaltons, so it's a pretty large protein. And I won't go too much into the immunology, but all you need to know is that interleukin-6 can upregulate your pro-inflammatory responses and also can participate in anti-inflammatory responses. And essentially, again, I want to quantify interleukin-6 in different samples. So interleukin-6 is a pleiotropic cytokine. So what does that mean? That's just a fancy word for interleukin-6 can do a lot of different things. It's a versatile cytokine. So essentially, it can affect inflammation in different ways. It's a part of many different inflammatory processes. So in some diseases, in autoimmune diseases and inflammatory diseases, different inflammatory processes can be dysregulated. And you can actually correlate this dysregulation of an inflammatory process with interleukin-6. So downstream, if you can quantify interleukin-6 in a lot of different cases, you can actually monitor the disease state of various patients. So if you look at this table that I produced, these are many different reported values for interleukin-6 in different disease state patients. So if you look at the column 1, you have the various inflammatory diseases. In column 2, you have the mechanism of detection. So just like us, other people made assays. Some of them are widely available, commercially available. You can buy them. You can use them to quantify different proteins. And you can see the third column is the sample, which is pretty self-explanatory. So that's the sample, the medium that was tested. And in the final column, you can see the range. So essentially, that's how the table is set up. So if you look at, for example, West Nile fever, the second column, interleukin-6 can be in a very wide range in the body. So this range is about 0 to 10,000 picograms per milliliter. So essentially, it's from undetectable amounts all the way to 10,000 picograms per milliliter, the nanograms per milliliter range. So what we're trying to do is essentially produce an assay that is both very sensitive, meaning it can detect a very small amount of interleukin-6, and has a wide dynamic range, so meaning it can detect a very large amount of interleukin-6. And to do this, it has to have a signal, whatever our signal is, that is linear over logarithmic concentrations. In other words, very large concentrations. So I developed several approaches to quantify interleukin-6, and I will begin with my first approach, the cap and release assay. So in this assay, essentially I developed these nanoparticles that you can see on the left. I won't go too deep into the synthesis. It was a process that took a span of months, but however, I'll describe what the purpose of the nanoparticles are. So essentially, I developed these hollow mesoporous silica nanoparticles. So what does that actually mean? That means there are pores on these nanoparticles that allows for a signaling molecule that we put in the center of those nanoparticles to eventually flow through. So essentially, I put a fluorescent signaling molecule called sulfurhordenine B inside of the silica nanoparticles. I then changed the surface chemistry of the nanoparticles to functionalize it with an amine group. And I did this for the purpose of changing the surface charge so that it has a positive surface charge. And the purpose of doing that is we have these
anti-interleukin 6 antibodies that are negatively charged. So this will allow these antibodies to bind on the surface of those nanoparticles. So those antibodies bind pretty tightly to those nanoparticles, sub-nanomolar range. However, antibodies bind to their target analyte typically in the nanomolar range. For those of you that aren't familiar with those types of concentrations, that aren't familiar with science, essentially antibodies will bind more tightly to interleukin 6 than they will bind to the nanoparticles. So what does that actually mean? When we have these nanoparticles that we produced and put in these Eppendorf tubes, in the presence.
So that was a portion of my doctoral defense. I hope you enjoyed it. So that'll give you a sense of how it works as an independent. You're an independent scientist working in a PhD lab, but you have an advisor who's guiding you, making sure that you have the resources available that you need to accomplish the goals and publish the papers that you'd like to. He or she is also there to make sure that the logic behind your experiments, your individual experiments, makes sense to eventually, accomplish the goal that you would like, making sure you have the right approaches. So that's a little bit about doing a PhD. So if you like that video, please go ahead and like and subscribe. Really helps out the channel. Don't forget to check out our TikTok and Instagram as well for short content. Don't forget to check out our website as well, thestemcenter.org, for it's a place where students can learn in a super interactive way. Also, if you're someone that's thinking about undertaking a PhD, you can go ahead and check out my website, andrewbrown.org. You can reach me in some way, shape, or form there. And if there's anything I can do to help, I would love to. All right, guys, that's the end of the video. Take care.